Ladies and gentlemen, welcome all to the 12th Karl Landsteiner Lecture of SEM in the beautiful festive hall of the Academy of Sciences. The lecturer this year is Yasmine Belkaid, and she is a director of the National Institute of Health Center for Human Immunology. Before we go into that part of the matter, let me briefly tell you a few things about this lecture series. If I can have the first slide, please. It's me? Okay, good. So, this lecture series that is also known among the cognoscendi as the gathering of the tribe, because we like to see as many friends, supporters of life science and biomedicine as possible, is centered around Karl Landsteiner. And the title of the lecture to Landsteiner has to do with the fact that we would like to, once a year, host the talk of a prominent scientist whose work is at the maximum influence to what we're doing. Somebody that is truly influential in biomedicine, and among all the several thousand people that would qualify, those that are actually having an impact on medicine. And hence, it's important because Karl Landsteiner himself, as you know, as the discoverer of the blood groups and also participant to discover of the rhesus factors, has, through his research, caused a tremendous advancement in medical treatment, in fact, in transfusion, and it's difficult to estimate the number of lives that were saved through his action, but are certainly in the millions. A little bit of the theme of tonight is also the fact that he was an émigré and was not entirely at his own will. It was for several reasons. Professor Beck, please, I'm sure we find a spot for you. I can't talk if I say you're here standing. Please do the job. And it is very important that the influence of Karl Landsteiner reminds us of the fact that even if you are into some sort of immigration situation, even if you are not under ideal condition, there will be a situation where you can find a good place to do the work. SEM is hopefully such a place. Very briefly, I know I bore most of you because you know what it's all about, but our center is clearly trying to achieve maximum innovation in medicine, and we are trying to improve healthcare by doing it. So that is a fairly focused kind of mission. We have an international team of scientists and medical doctors, and we are trying to do the most basic, free-minded kind of research that you can do within the context of the medical world that we're privileged to be in, as the place is in the center of one of the largest research hospitals and campuses in the world, which is all around us. We are focusing on different kinds of disorders that have to do with immunity, metabolism, but also cancers. And we think that we have quite a unique mode of super cooperation in the diction of Martin Novak, the mathematician, because we are going beyond what is customary in trying to work together to achieve greater goals. And this super cooperation goes beyond the way we interact among each other. It also expands to the way we interact with society and the way we are trying to make bridges between different areas. It's absolutely key to our mission to train young scientists and medical doctors, and we do this with as much heart and brain as we can. And we are contributing to the constantly changing paradigm in medicine that is bringing a type of medicine where prevention is going to be increasingly important, and also the ability of being participant to your sort of the collective medical wisdom and health. And that is a great challenge that includes a lot of other people beyond the group of scientists that are SM. Very briefly, we are about 180 people, 70 of which are PhD students. We have quite a few ERC grants. I welcome the former president of the European Research Council, Helga Novotny, who is one of the persons that inspires me. In fact, we can start with a nice applause for her. Thank you, Helga, for joining us. Your mentoring is important to the community. And so we are very proud of the fact that we have quite a good number, not yet quite as high as we wanted, of ERC grant, but also of grants such as the starting grant from the FWF that is very important, and the similar grants that are called New Frontier from the Academy of Sciences. And most of the people except me are young in the institution, and we have some 42 nations, so we're very international. And this is all I really wanted to tell you about SEM, except that we have now the new research report that is outside. Please look at it. It is the work of many, many people, including Wolfgang Däuble and Eva Schwenk, who are here. But also we have, also take it. If there's not enough, you can register your name. We can send it to you. But also there is a little booklet called 10 Years of SEM, so we have it in 2 languages. And this booklet is funny because now with an app that you can scan, that you can download, it's augmented reality SEM, you find it on the App Store. You will be able to look at augmented reality features from this booklet. So it transforms from a simple booklet into something magical, where people start speaking, things start moving, and so on and so forth. So if you have a young person at home that is considering adventuring into research and science, please, maybe this booklet is a way to get that person interested, because it is in true one of the first such examples. So Yasmine's talk is too good to not pay attention, but after you may download the app and have fun doing it. So both are outside for you. Very briefly, the previous Landsteiner lectures are too many to mention in their entirety. For sure, they all had to do, like John Korean, with the ability of making something that had an impact on therapy. Vishwadixit Kari Stefansson, that was almost killing a journalist, you remember, because he was challenged on his decode ethics. Helen Hobbs, fantastic talk. George Daly on stem cells. Ruslan Medzitov dancing with us at the violin of the Crimean player. Hans Klever's beautiful movies. And David Sabatini telling us the intersection between metabolism, growth, and many decisions that are based on this in the cell. Laurie Glimcher, a beautiful new approach on cancer immunotherapy. 2 years ago, we had Manu Charpentier, who is not new to this group and audience, about the revolutionary CRISPR technology. And last year, we had Jay Bradner tell us about a new innovation that happens in the interface between chemical biology and drug discovery, and in particular about a drug that is modifying chromatin. So what we would like to do now is to invite 2 musicians to sing us and play us 2 songs about 2 Viennese composers that were active between the 2 wars and have composed some very nice melancholic springtime ode to love, wine, and Vienna. And they both ended in emigration. They were successful, and they are in many ways the type of people that we think we should be thinking of more often. Their names are Fritz Spielmann and Egon Neumann, and we'll hear Wien tausendmal schöner, wir tausendmal schöner from Fritz Spielmann, and we'll hear Bissel Liebe, Bissel Wein of Ego Neumann. And the two musicians that will make this dream happen, because you see that you'll be dreaming as soon as they start playing. At the piano, Bela Coregni, a very well-known piano player who has been at the legendary Broadway piano bar and has played with Helmut Qualtinger and many, many people. Those of you who are night owls and older than 50 are likely to have met him more often than some of the young people. But he is now into a new team with a fantastic soprano, Ethel Merholt, and they just had a release of a new CD or a new vinyl record. And the singer that I already mentioned, Ethel Merholt or Merholt, she studied in Vienna and in other places. She sang also for the opera. She is, since now a few years, has entered this collaboration with Bela Coregni. So I'd be very happy if you turn off your mobile phones so that we can welcome Bela Coregni and Ethel Merholt for their musical introduction. Thank you very much. Thank you very much. It's absolut fantastisch. Ja? Danke sehr, danke sehr. Bela Coregni and Ethel Merholt. Ja, danke. Danke. Wiedersehen. So, and now to our speaker. It's a fantastic pleasure to introduce Yasmine Belkaid to you. A person that has reconciled us with the zoo that is in us and around us, and has helped change the way we understand the relationship between the bacteria and ourselves. Very briefly about her CV, she was born in Algeria, where she studied both her first 2 degrees, and then went to Paris to Institut Pasteur, where she was with Geneviève Milon, I understand, and where she fell in love with immunology, and in particular how the body is dealing with parasites and bacteria that are living with us, and we are not fighting them, but we have a way by which we adapt, or the sort of parasite or the bacteria have a way to make it possible. After that, she went to the US, where she remained since, both at the NIH, then in Cincinnati, and then back to the NIH, where she assumed different responsibilities. She did postdoctoral fellowship with David Sachs, and she has been also there a pioneer of understanding the relationship between sort of bacteria and sort of the mucosa that we have in our bodies, as well as then how it can go wrong in disease. She's now the NIH director of the Human Immunology, as we already said, and has more than 2,000 peer publications. Of all the awards that she has obtained in the last several years, I will mention the fact that she was elected to the US National Academy of Sciences, as you know, something that is a pinnacle in recognition, and it's something that we all would greatly aspire to. But also she obtained a few very important prizes, many important prizes. I would like to mention the Emil von Behring Prize 2017, because it's exactly in the area of the interface between bacteria and human health that she will speak about. And some of you may have seen the TV series that was broadcasted not too long ago with Koch, Behring, Ehrlich in Berlin at that time. Had there had Yasmine, would have been Yasmine, we would have had an earlier role for a woman. As you have realized watching the not badly done TV series, it was a man's world where women were there to assist the work of the professors, of the aspiring professors. And there was much more political, even more political sort of clout about science that we have now. So even if we complain about situation these days, and I think that those days were not avoid or devoid of influence. She also got an important prize from the Sanofi Institut Pasteur in 2016. But for us scientists, a few things are very easily recognizable. One is that she's been an organizer of Keystone meetings and Gordon conferences, some of the best series of conferences in the world. But also she's an editor of Cell and Science, and so most of us will be well advised to be as nice as possible to her, as she has that big trump in her cards. Now, bacteria keep to be one of the leading cause of death worldwide, and we have a program studying immunity at our place. And Sylvia Knapp has a major interest in, for example, the bacteria in the lung and how they are affecting the immune responses and how they can co-live with the resident immune cells. But a lot of what we know about how the cell can, how the system, the body can deal with bacteria is really work that Yasmine Belkaid contributed to. First, understanding the role of T cells, understanding the role of how you build tolerance to certain, let's say, allergens, both in terms of food, but also in terms of sort of parasites. And then, most importantly, in trying to understand how the interplay between this microbiome and the different surfaces, the mucosa, happen over the immune system, so that we are keeping the good bacteria and trying to fight off the bad bacteria. Our knowledge of this interplay between bacteria and the body, and in fact nutrition, is really heavily contributed by Yasmine's work. We are sort of very, very grateful that despite her tight schedule and her commitments, she has accepted our invitation to give this lecture. We are entirely sure that she will be inspirational also for young scientists and young persons that are looking always for role models and people that we can look up to keep us do research with is fundamental. And we are sort of hoping that this visit will trigger some sort of love affair of her and Vienna. We tried with music, we tried after with wine, and certainly we tried today at SAM with science. And perhaps if we're successful, we will see a sort of more of her in a more sort of regular fashion. We have a little present that is used to remind us of. So please welcome Yasmine Belkaid. It's in this way or this? So this is the commemoration this way of the Landsteiner lecture for your library. But we also have something that is sweeter, and that is the famous Sachertorte. Between the two, we hope that you will have a fond memory of this place. I can bring them to you. So thank you very much, and we're looking forward to your lecture.
Thank you very, very much for this wonderful introduction, for the invitation. I mean, it's really a true privilege to be here today, standing in front of you. And I'm so Vienna. I mean, definitely it's a gorgeous place, and the welcome has been absolutely wonderful. So today, for the next few minutes, I'm going to discuss with you some of the work we're trying to do to understand this complex interplay between our immune system and the multitudes of microbes that are born with us and are colonizing every single body surface. So this is not a new concept, and in fact, the grandfather of the microscopes a long time ago, uncovered in his own saliva this multitude of microbes, and you can actually see his beautiful hand drawing of these microbes. So it was really the first discovery that we're not alone, and all body surfaces were actually colonized by a highly diverse group of microbes. And of course, we have gone a long way since this first observation. And the work that was done collectively by numerous scientists in the world has actually identified that very soon after birth, all of us are colonized by highly diverse microbes. These are not just bacteria, they're also viruses, fungi, protozoans, all the things that you have learned to fear are actually born with you and colonizing all the body surfaces. These microbes are actually highly numerous. It is believed, and they are at least 10 times more numerous than our human cells. And it is believed that there is 100 times more genes, and probably more genes expressed by these microbes than our own genes. So just to give you a small schematic, when you have an illusion that you are a very important person, remember that most of your genes and most of the function of your body are actually carried on by these microbes that live with us. So this is an enormous potential for adaptation, change, and function that we're actually carrying in our all body surfaces, GI tract, skin, and so forth. So the understanding then this microbe are actually positive for us is also not a new concept. Meshnikov actually, long time ago, had uncovered the link between bacteria, at least the one coming from milk, and human health, and was actually able to propose a concept of beneficial microbe as a way to prolongate life. But really, the concept that microbes were really gatekeepers of health really came in a more impressive way by a German scientist that really showed that actually lactobacilli in the vaginal ecosystem was extremely important to protect women against candida infection. So it was the first discovery that microbes that live in certain body surfaces were playing a fundamental role in limiting infectious disease. So of course, we have today, with the emergence of novel technology and really the enormous enthusiasm that scientists have collectively developed in trying to understand the interplay between the microbiota and health, over the last 10 to 15 years, a large body of work, both experimentally but also in the context of clinical settings, had actually discovered that the microbiota play a fundamental role in all host physiology. And some of these findings are actually summarized here. Of course, microbes that live in our GI tract are important because they can help us digest, they can actually help us metabolizing vitamins and biles and so forth. The microbiota can also play a fundamental role in host physiology. Pioneer work that was done by the group of Jeff Gordon showed that if you take the microbiota of an obese animal, you can actually transfer obesity just by changing the microbiota composition. And this really opened the door to numerous research really linking the microbiome to host metabolism, and really is really a fast developing line of research linking the microbiota to type 2 and type 1 diabetes, for example, and so forth. More recently, several groups have uncovered an unexpected link between the microbiota and the nervous system. And at least experimentally at the moment, it has been shown that different microbiota composition or relationship with the microbiota can affect the behavior of animals. This is really an emerging line of research, but propose a really fundamental connection between this microbe and the nervous system. And what we and others have actually uncovered over the last few years is this fundamental link between the microbiota and the immune system. And that's what I'm going to discuss with you today. So work that collectively was done by our group, but also many others, found that the microbes that live initially in the GI tract, and I'm going to first talk about the gut microbiota to talk about other microbes later, play a fundamental role in all aspects of the immune system. Which means that for your immune system to develop properly, you really need encounter with the microbiota, and more particularly with elements produced by the microbiota. For example, hematopoiesis, which is actually the ability of your body to generate cells that are important for the immune system, is controlled by the microbiota. And this was actually the first discovery that microbes are not only able to act within the site they are colonizing, the GI tract, but also to act distally in all the other organs, and in particular in the bone marrow environment. The microbiota can really act as an adjuvant, and I'm going to talk about that a bit more during my talk. This is really a natural adjuvant of the immune system, allowing the development of antibody responses or T-cell responses that can combat infection. And finally, the microbiota is also able to control local immunity, which is able to constantly tickle the local tissue responses, allowing cells that are important for the immune system to function when they enter in the site that is colonized by the microbiota. Because of this profound alliance, the microbiota, of course, has been shown to play an important role in many aspects of the immune system, and this can be in the context of a response to infection, but also in the context of vaccine responses. And more recently, it has been really an explosion of understanding of a link between the microbiota and immune therapy. And I'm just going to summarize a couple of those findings. So what has actually been shown initially experimentally using animals and then subsequently in the context of clinical trials is that the quality of the microbiota in the gut of an individual can be predictive of the ability of the person to respond to immune checkpoint therapy, which is a new form of therapy that is actually revolutionizing the field of immunology and cancer therapy. So the intensity of an immune response in the context of tumor can actually be influenced by the presence of the gut microbiota. It's a quite unexpected alliance that is now opening the door to a whole new association and really helping clinicians to now begin to integrate this new component to try to understand why patients can actually respond or not potentially to certain treatment. Of course, all alliances can actually go in a negative way, and I'm not going to talk that much about this negative way during my presentation today, but nonetheless, we should keep in mind that a dysregulation of this profound alliance can actually have negative outcome. There is, of course, a link between the microbiota and inflammatory disorders because they can engage the immune system, and that has been really beautifully illustrated in the context of inflammatory bowel disease, for example, in which it is now clear that part of the disease is actually sustained by aberrant reactivity against the microbe that live in the GI tract. The microbiota, as I mentioned before, can regulate host metabolism, and in some cases in a negative way by promoting obesity and metabolic syndrome. And the microbiota can also promote the development and intensity of autoimmune disorders. So really maintaining this alliance in a peaceful form of interaction is fundamental for the maintenance of human health. So our own interest to try to understand the link between the microbiota and the immune system came from the very simple observation, that is that all pathogens, or what we consider as pathogen, access the host via tissue that are naturally colonized by the microbiota. The first site that are infected, in which the microbes are either infecting or entering the immune system, will be the lung, the skin, the GI tract, which are all naturally colonized by highly diverse microbes. Which means then this first form of interaction of the microbes that can infect with us is through this internal competition with this microbe that live in all body surfaces. And of course, there's an enormous layer of complexity in this form of interaction, and I'm going to summarize some of the findings of the field. One thing that is also important to remember when we try to actually in a simplistic manner, divide the pathogen versus the microbiota is the fact that the microbiota of all of us at any given time contain microbes that can potentially become pathogens. And this will be the case, for example, of highly resistant antibiotic resistant pathogens. This could be the fact of many microbes that can, in certain settings, become aggressors. And these microbes, under normal circumstances, are held in check inside our gut or in our skin by the action of the microbiota against these invaders. So there is an enormous amount of competition that limits the expansion of these microbes. So some of these examples of this competition can actually, of this effect of the microbiota in the context of pathogenic infection, can be summarized in this slide. So, for example, the microbiota has been shown, as I will discuss today, to promote immune responses. And in this example, for example, is shown antimicrobial peptide production. The microbiota can also have metabolic products that can in themselves affect directly pathogens. The microbiota can actually compete for nutrients, which is actually a very important source of defense because they're consuming nutrients, limiting the colonization of microbes. And finally, they can have direct toxicity. In fact, the microbiota is an enormous reservoir of molecules that can have antimicrobial function. So really, there is a whole new area of research trying to identify this molecule, because within our gut, we have our natural drugs that can actually combat infection, or natural antibiotics. So a lot of research today is aimed at really uncovering those molecules, because we believe that this may be a more physiological way to promote a defense against pathogens. So an example that can really, in a very spectacular way, reveal the power of this microbe in protecting us of an infection is what most of the clinicians know very well. If a patient is actually treated with a large spectrum antibiotic, this patient can actually develop very severe infection. And this is due to the fact that you have removed all the beneficial microbe of the gut, and in these circumstances, those rare microbes that live within your gut can actually expand and really dominate the gut environment. And this can lead to very severe consequences for patients. This will be the example, for example, of Clostridium difficile, that is really a serious problem in the context of clinical settings. So this understanding led to a form of really very, very simple therapy that is aimed at replacing the microbiota, and this actually is fecal transplant, that in the context of certain infection, especially C. difficile, had really transformed the treatment of this disease. So the approach is extremely simple. What happened, the patient is replaced with a novel microbiota, and this actually allows to prevent colonization of with C. difficile or other antibiotic drug-resistant pathogen. So this form of treatment really was the open door and a proof of concept, and you can actually treat a patient by changing the microbiota composition, and in some cases really limit very severe infectious disease. And really, as I mentioned before, this has really revolutionized the treatment of certain infectious disease and led to a complete treatment of an infection that was not previously possibly treated. And again, this approach of really trying to find the right microbiota to treat disease is now extended not only in the context of infectious disease, but in the context of other syndromes, in the context of inflammatory bowel disease, but also in the context of immune checkpoint therapy, in which a large number of scientists today are trying to identify the right microbe that could be potentially provided in the context of treatment. So can we actually rectify the microbiota composition of a patient to boost the immune system in a way that responds better to cancer treatment? So I'm just going to give you, since the science talk, I'm going to give you a few examples of actually how we came to this observation and who were actually the players in identifying this function. So our understanding that the microbiota can boost the immune system came from the work of a graduate student in the laboratory, Jason Hall, who did a very simple experiment. What he did was actually to treat animals with antibiotics and remove most of the microbiota in the GI tract. And he was able to show, using this very simple approach, that removing the microbiota in the gut also limited the ability of the tissue to develop immune responses. And in this case, in particular, to promote responses such as interferon gamma and 17, which are molecules extremely important to control infection. And as such, the animal became highly susceptible to the infection. So really removing the microbiota had an effect on the ability of the host to mount immune response to a pathogen. At the same time, work that was done by Dan Littman uncovered a very fundamental link between different microbes and the ability to push the immune system. And the work he did with a very canonical microbe in mice that is called SFB was to show that certain microbes are able in a very specific way to push certain kind of immune responses. And in particular, in this case, IL-17 response that is very important for gut health and tissue barrier function. So these really together showed that the microbiota was important to promote immune responses, and then defined microbes in the gut microbiota were better than others at pushing the immune system. But this is not just bacteria. As I mentioned before, we are colonized by highly diverse microbes, not just bacteria, but also virus and protozoans. Work that we done in collaboration with Miriam Mera a few years ago uncovered, for example, that protists, which people believe are actually usually parasites that can live in our GI tract, are also very good at promoting the immune responses and change the way the host develop response to a pathogen or develop pathology. So a protist can also influence the immune system. So is a virus. Work that was done by Ken Cadwell at NYU showed that virus in some cases can be sufficient to also push the immune system. So remember that we have this whole diversity of microbes. All of them have co-evolved with us as a way to push the immune system and to promote antimicrobial defenses. But as I mentioned before, the relationship between microbes and the host can in some cases have negative consequences. So it has been shown, for example, to the work of Richard Grancis, that in some case a worm to infect the host require the interaction with the microbiota. So the worms that are actually able to infect humans are present as eggs that need to hatch, and this hatching process can be mediated by the encounter with the microbiota product in the GI tract. Work by Pfeiffer at UT Southwestern showed that certain viruses, and in particular viruses that can be acquired orally, are also requiring the microbiota to become infectious. So in some cases, the microbiota can help a pathogen to actually invade the host. So there is actually, of course, in some rare case, negative consequences. And importantly, the microbiota in some cases can also become an agent of pathology in the context of infection. As I mentioned before, the microbiota push the immune system. When it does in a way that is excessive, this can have negative consequences and enhance pathology in the context of infection. So, in summary of what we know about host microbiota interaction, work that we have done, but also many other people in the field, has actually uncovered that virtually all aspects of host pathogen interaction can be controlled directly or indirectly by the action of the microbiota. So as I mentioned before, transmission of a pathogen can be influenced by the microbiota in a positive or in a negative way. Response to a pathogen can be influenced by the microbiota because for the immune system to function, it needs its constant stimulatory effect of the microbiota. As I mentioned before, in some cases, microbiota can push pathology. And the way it does is because inflammation can change the composition of a microbe in our gut and can allow the emergence of microbes that are more aggressors. So in this case, they can contribute to pathology. And finally, I'm going to briefly discuss today, the microbiota can also, in some cases, promote a fundamental aspect of tissue physiology that is called tissue repair. So one question that we really begin to address more and more over the last few years is really the nature of the immune response we develop against the microbiota. So most of what we know as immunologists trying to understand host defense and all of what we know about how our immune system works came from the observation of how the immune response develops in the context of an infection. So when you have flu, when you have a GI tract infection, what is the nature of the response you develop against? Because of course this is what we want to understand in the context of development of vaccine, in the context of immune defenses. However, most of what our immune system does on a daily basis is not to deal with pathogen, but it's constantly sensing this non-invader, this multitude of microbes that are living in every single body surfaces. So what is the nature of this really remarkable dialogue, and what are the regulation and rules of this form of immunity that we virtually know nothing about, because all of us have been so busy trying to understand the aggressors. So in other words, I'm going to actually summarize where we were a few years ago and where we think we are today. So where we were a few years ago, we had created this dichotomy, pathogen in one hand and the microbiota in another one. So we believed, in a relatively naive way, that our immune system was only here as a means of defending ourselves and only alerted in the context of invasion, tissue damage, and inflammation. And what we believe is the way we dealt with the microbiota is we basically ignore them. They were outside. They were outside of our gut, outside of our skin, outside of our eyes. And we developed this naive understanding that it was ignorance. And in fact, this is not the case. If anything, the immune system is constantly seeing these microbes. And although they are not invaders, although they are living outside, our immune system really is designed to constantly probe and sense these living creatures that are living everywhere in our body surfaces. So what we decided to explore a few years ago is the nature of this interaction and what could be the rule and what could be the feature of this different class of immunity that we really need to understand if we want to understand host physiology and human health. So the tissue that we decided to explore a bit more over the last couple of years was the skin. So most of the work we did for a few years ago was in the GI tract, but we became really interested by the skin for obvious reasons. It's a tissue that is highly accessible, a tissue that is of course the target of numerous inflammatory disorders, a tissue target also of cancer, but also a tissue that is a target of numerous infectious disease. And the skin, although something that we think less about, is also naturally colonized by highly diverse microbiota. So in the human skin, and this was done by the work of Julie Segre at the NIAID, if you sample 40 different sites in our own skin, we actually have 40 different communities of microbes, because what those microbes do is really feed on the unique nutrients. And our skin is actually quite diverse. So each of these body parts are actually colonized by distinct kind of microbes. And these microbes can, of course, be affected in the context of inflammatory disorder, but under steady states are relatively stable. They are not only bacteria, but they're also fungi and virus, and it is believed that they may be even more diverse than what we have in our GI tract. So work that was done a few years ago by another talented graduate student, Shruti Naik, uncovered that the skin, despite the fact that the biomass of this microbe is very low, was still controlled by the microbiota. And what she was able to show that in the absence of skin microbiota, the skin immune system was not able to control infection, and this was actually occurring via the ability of the microbe to promote local responses against infectious disease. Just a couple of words about the skin. This morning I had a discussion with students. You can actually shower yourself. They are stable, they are there. So they are mostly present within deep structure in the skin. They're not at the surface. They live in our hair follicle, in our sweat glands, in our sebaceous gland, and they really establish a very stable colonization. So they're not washed away every time you go swimming, don't worry. So having uncovered this association between the microbiota and the immune system of the skin, we decided to go into a quest. And the quest was the following one. Can we begin to identify microbes that are uniquely able to push the immune system? And this could potentially open us the idea of manipulation of the immune system via the microbiota transplant of the skin. So the approach we use is very simple. We collect primary isolates from numerous patients. We obtain them from patients that suffer from melanoma. We have them from patients that suffer from autoimmune disorders, various inflammatory disease, psoriasis, atopic dermatitis, but also from healthy donors. These microbes are identified, they actually are sequenced, and then we actually probe for the ability of this microbe to engage the immune system. So the skin at steady state condition contains a very diverse population of lymphocytes. We apply the microbe at the surface of the skin. There is absolutely no injuries. It's just a very simple painting in which we don't create any kind of abrasion. We just basically colonize the animal, and we look at the ability of this microbe to engage the immune system in a specific way. So this actually allows us to, when we have identified microbes that have a unique ability to push the immune system, to look at how this microbe can influence different states. We create different kind of inflammatory settings. We're looking in the context of cancer. But more importantly, what we're trying to do, which is really the bottleneck of the field at the moment, is to understand the mechanism by which this microbe engage the immune system, both at the level of the microbe and at the level of the host. So collectively, this led to, over the last couple of years, to work that was done by a few people in the laboratory, uncovering a remarkable association between unique microbe and the ability to engage one subset of lymphocyte and not another's. And what is important to remember for the people that don't think about immunology all the time is each of these immune cells have different function and can help you to control different kind of infection or different kind of inflammatory settings. And what we found is actually the microbe themselves are uniquely able to engage one cells but not the other one in a very, very surgical manner. So I'm going to just give you one example of that to illustrate the contextuality of this role and how this could actually in some cases be influenced by nutrition. So Corynebacterium is a bacteria that live in all of us. All of us at any given time have multiple strains of Corynebacterium in our skin. They are very gentle microbes, they are not creating infection, and all of us are colonized. So what Vanessa Ridera was able to show is that Corynebacterium, despite the fact that it doesn't cause inflammation, has a unique ability to promote the accumulation and activation of one subset of cells that is called the gamma delta T cells. That is a very important population of lymphocytes. And this was true for all the microbes that belong to this Corynebacterium family. What she found is one microbe from Corynebacterium family was not able to do so. And this microbe is a natural mutant that is actually lacking a fundamental molecule in the cell envelope that is called mycolic acid. And this really pointed us to the fact that this molecule may be what mediates the dialogue between the microbes and the immune system. So we took the bacteria and generated a mutant that can no longer produce mycolic acid, and indeed, not being able to produce mycolic acid abrogated the ability of this bacteria to push gamma delta response. So this is just a very small example to show you how in some cases by comparing numerous strains you can begin to identify the molecule that are utilized by the microbe to communicate with the immune system. And this is a step forward in the context of going to therapy, because in some cases we could imagine that transferring a microbe may not be a viable option, but if you understand the molecule they're utilizing, we may be able to push immunity in a very specific way and creating a new line of adjuvant that is designed based on the way this microbe have co-evolved with us. But the point I wanted to utilize by this very small vignette is the following one. So what we wanted to do is change the nutrition. And the reason we did that is because we know that there is a dramatic link between obesity and inflammatory disorders. And Corynebacterium is also a bacteria that loves and thrives on lipid metabolism, so consuming a lot of lipids. So the experiment is very, very simple. We apply the microbiota, the Corynebacterium, at the surface of the skin of an animal that is fed a regular diet, and you can see that there is absolutely no inflammation. But if you actually put the animal on a high-fat diet, the same microbe now is causing inflammation. So it's a very small example to show you that even our own microbe, that our healthy partner, if you change your diet, you can transform these allies in enemies. And they can now be sufficient to cause inflammatory disorders just by changing the lipid content of your diet. So when we think about the dramatic emergence of inflammatory disease in high-income countries, we may want to think about the way now our healthy partners may actually be perceived by the immune system. And we believe that actually this may have an important consequences in the etiology of a large number of inflammatory disease, and we are now trying to uncover the mechanism underlying this transformation. But next time you go in vacation and eat too much lipid, maybe think about it a bit more. This can have consequences. So the point I wanted to make with this very small vignette is to remind you that the ability of a microbe to cause disease can be, in fact, contextual. And although we try and we want to actually separate the bad from the good, life is a bit more gray. And there is a lot of circumstances in which our healthy partners that we have co-evolved with to help us in many different ways can become our enemy. And this can actually happen in the context of genetic predisposition. If you have, for example, alteration of gene expression that allows you to maintain regulatory responses in our body, now you can actually you can aggress those microbes. In other way, if you can also have genes that are important for maintaining barrier structures, this actually can have consequences because this microbiota can enter into the immune system and become inflammatory. And as I mentioned before, just a change in diet can be in some cases sufficient to make this relationship detrimental. So microbes travel in a continuum, and the circumstances by which we are genetically wired, or nutrition, or different inflammatory setting can change the way we actually sensing microbe and transform this relationship. So for the next few minutes, I'm going to talk about some ongoing work in which we're really trying to understand what is unique about this kind of immunity we develop against the microbiota. And this is done and has been done in the laboratory by a very talented team of postdoctoral fellows, Ollie Harrison and John Linham. And what they did is actually following up on some of the observation we had a long time ago, then Staphylococcus epidermidis was able to also engage one single cell subset that is called the CD8 lymphocyte, that many of you do know because those cells are important for antiviral defenses. So the experiment that we had done a few years ago is the same principle, very simple. What we actually found is actually when you apply Staphylococcus at the surface of the skin, it can actually promote a dramatic accumulation of this population of cells that is called CD8 T cells that can produce cytokines that I mentioned before are important for antimicrobial defenses. What was really intriguing is that this microbe was able to do that, but it was really the only one that induces CD8 response. Staphylococcus epidermidis, once again, is present in all of us at any given time. This is not a rare microbe. All of us carry this microbe at any given time. What was also quite dramatic is the fact that there is absolutely no pathology. This response to the microbiota was really important, very significant. It was a lot of CD8, but this did not cause any inflammatory responses, which is different from the way we sense a pathogen. Every time you sense a pathogen, you develop inflammation. So responding to the microbiota occur without inflammation. So what Shruti at the time was able to show is that was a very beneficial event for the immune system. So what she found is that within the hair follicle in which the microbiota lives, a subset of dendritic cells, which is very important to initiate the immune responses, is able to enter the hair follicle, capture the bacteria, and migrate to the lymph node in which immune response can be initiated. Within the lymph node, these dendritic cells are able to stimulate this population of CD8 T cells that migrate back to the skin, and because they can produce this molecule IL-17, they can actually stimulate the activation of keratinocytes. And now the keratinocytes are producing a lot of antimicrobial peptide that are extremely important to control infection. And when you come back with another infection, now the animal is actually better protected. So immunity against the microbiota can actually, in a very indirect way, promote antimicrobial defenses by enhancing antimicrobial peptide production at the surface of the skin. So work that was subsequently done by another graduate student laboratory, Alison Bird, was able to show that this association was actually not all the staph epi. So she utilized a large number of strains that were fully sequenced and annotated by the group of Julie Segre, and was able to show that the ability to promote CD8 responses was actually restricted to one group, one clade of Staphylococcus epidermidis. So people that are not bacteriologists here think that actually all bacteria from the same species are the same. This is not true. In fact, within one species, there is an enormous amount of diversity. And to try to understand the link between bacteria and the immune system, or bacteria and host physiology in general, in some cases, we may have to go at the level of the strains or the clade to try to understand mechanism, which is really an additional challenge for the field. But what she was able to show is that this A20 clade was very important in pushing the immune system, and in particular CD8 responses. And looking at the large survey of healthy donors, she was able to show that in human, most healthy donors really have this A20 clade, and then this A20 clade was actually diminished in the context of atopic dermatitis. So this is not approved, but is a beginning of association. Then this clade may actually offer an advantage for the host, and what is actually diminished, this may in some cases promote inflammation. So a couple of words for the immunologists in the audience. We try to actually understand what was really seen by the immune system. So to be seen by the immune system, you need what is called an antigen. A lot of you have heard about antigen in the context of vaccine. An antigen is an element in the bacteria that can be seen by the immune system. It's a very specific code that can be seen. So it is strange, because you have probably seen news and you know that all of us, a lot of people work on the microbiota, then we still don't really know the antigen expressed by the microbiota that are seen by the immune system. And when you think about this enormous amount of microbe living everywhere, we really virtually know nothing about how they are seen. So, and this is actually just a small summary of the 3 or 4 antigen in regard to the billions of putative antigen that could be present in the microbiota. So we really need to really understand the antigen scene. So this actually quest was started in a laboratory by John Linham that decided to begin to, if we want to understand the nature of this interaction, we need to understand the antigen that are seen by the immune system. And what could be the feature of this response or mechanism. And I'm just going to give you a very short summary. So what he was able to show is that in a very strange manner, a lot of the antigen that are seen by the immune system are seen by what is called non-classical MHC molecules. So in humans, you have actually, and in all mammals, you have what is called classical MHC and non-classical. And most of response to pathogen are in the classical MHC. But what we find is that a large number of antigens seen by the immune system are organized by this other mysterious class of non-classical MHC molecules. So it appeared that even the molecules that are utilized by the immune system to see commensals is actually different from the molecules that are utilized in the context of an encounter with a pathogen. What is really intriguing in this specific example is only one of them, is that the molecules that are seen by the immune system are N-formulated peptides. N-formulated peptide is a canonical feature not only of bacteria, but also of the mitochondria. And some of you may actually know that the mitochondria that live in all our cells is of bacterial origin. So it's quite a fascinating point to imagine then a system that is in place in our immune system to deal with our own self-antigen, mitochondrial derived. Maybe also a system that helps us dialoguing with this exogenous microbe present at all body surfaces. But the point I wanted to make by showing this very small vignette is that clearly some of the molecules utilized by the immune system to see the microbiota may not be the same molecule utilized in the context of an infection. And to finish, I just wanted to show you some unexpected data that we obtained recently, in which we started to compare lymphocytes, which means that, of course, the cells responsible for attacking infection in the context of an infection, or lymphocytes generated against the microbiota. And we just looked at the gene expressed by those cells. But what was really, really strange is when we looked at the signature expressed by the one generated to the microbiota, is that a number of genes expressed by these cells look like genes that are involved in tissue repair and remodeling. And this is not something you see in the context of a T cells generated to pathogen. Those cells are mostly aimed at attacking. So this actually told us that maybe there is another function by those cells, and maybe they could also promote this fundamental tissue repair process. So the approach is extremely simple, in which we basically apply the microbe and look at the ability of the host to regenerate and repair after in the context of a small biopsy. And we utilize either the strain that can promote the CD8 responses or strains that do not induce the CD8 responses. As I mentioned before, we have those different microbes. And the first thing that you can see, and I hope you can see on this slide, is that the T cells specific for the microbiota have a really remarkable tropism. If there is a very small biopsy and a very small injury, all the T cells against the microbiota are able to redeploy themselves and create a ring at the edge of the wound. So there is something really unique about these immune cells in which they are clearly able to very quickly create this protective armor at the site of injury. So we're now looking at the function of these cells, and in particular one we wanted to understand was, are these cells able to promote repair itself? And this is what you can see here. You can see that indeed, if you utilize a small biopsy and have these bacteria that can promote repair, they can actually accelerate the wound healing compared to a strain that does not induce CD8 T cells. And this actually suggested that really the bacteria can in some cases not only enhance antimicrobial defenses, but may be also able to promote an extremely important aspect of life, which is repair of the host. And this is basically another way to approach this question, in which what we look at is the proliferative response of keratinocytes. So when you have any kind of small injury, any scratch on your skin, the way you repair is allowing this keratinocyte to proliferate, and they then migrate to create what is called an epidermal tongue. And this allows eventually the closure of the wound. It's a very fundamental process that is highly regulated, but you can also quantify it in a very precise manner. And again, you can see that if the animal has a bacteria that contain these CD8 inducers, you accelerate this repair process. And if you utilize animals that can no longer develop the CD8 responses because they're lacking this non-classical MHC molecule, now this protective effect is abolished. So this actually is to illustrate the fact that immunity to the microbiota is a really complex form of immunity that is not just aimed at antimicrobial defenses, but also aimed at controlling tissue physiology and can actually promote tissue repair. So in summary of what I have done, I'm going to just show you this here. We found that the microbiota can push the immune system in many different ways. One of them is, as I mentioned before, to promote the production of cytokine in tissue that are so important to allow the cells to function, allowing them to control infection. But the microbiota can also be seen, and it's something that I think is a new understanding of our relationship with the microbes. They are constantly seen by the immune system. And although they are not invaders, our immune system is designed to constantly probe and collect those microbial products or those microbes themselves. In this example I showed you, dendritic cells can collect the microbes, can go to the lymph node, induce those CD8 responses that can go to the epidermis and enhance antimicrobial defenses. So this adaptive immunity to the microbiota can provide this protective layer. As I mentioned before, is a response that is non-inflammatory, that is very physiologic. It happens in absence of any inflammatory responses. And in the example I gave you, can be mediated by molecules that are traditionally not utilized in the context of host-pathogen interaction. And as I mentioned before, this is a complex form of immunity. This is also a complex immunity that can not only enhance antimicrobial defenses, but the cells themselves can actually promote other physiological responses, including tissue repair, because they contain numerous molecules, such as amphiregulin, MMPs, and others that are so important for tissue remodeling and repair. And we have actually early evidence showing that the microbiota in the skin is also able to regenerate nerve terminations and also to influence pain sensation, really showing that there is a profound alliance between this microbe living in our body tissues and the promotion of host physiology and tissue response at large. So in conclusion, this is a very young field, and I think although there is a lot of news about the microbiome, we still understand very little. But this is really a gold mine of discoveries for all of us as scientists, because for the first time, all of us can begin to communicate around one language that is the microbiota. And it's the first time I see a lot of collaboration between microbiologists, immunologists, clinicians, chemists, people working on small molecule discovery. And together we really are beginning to tackle and decipher and uncode this kind of remarkable dialogue between this diverse microbe and host physiology. And I really truly believe that by continuing to exploring the mechanism of action of this microbe lies extraordinary discovery in terms of medicines and really the natural medication, the natural elements that can push our immune system in a way that is highly physiological has co-evolved with us. So learning from these microbes in a very rigorous way may really open the door to novel therapeutic approach that are aimed at combating infection, promoting tissue regeneration, enhancing immunity in the context of cancer immune checkpoint therapy. So I think this is really an exciting time for the field of research, and it's a new understanding that we are not alone, that we need to respect our microbe and learn from them if we want to develop a more physiological way of addressing medicine. So the most important slide is really the people that have done the work. I've been extremely lucky to being joined in my laboratory by numerous fabulous scientists, both PhD and postdoctoral fellow, and also over the years to have many collaborators that have opened the doors to many different fields of investigations. I need to thank all of them, but for the purpose of this talk, I'm just going to thank in particular John Linham, Oliver Harrison, Jason Hall, Shudenaik, that have highlighted the work of, and of course all our numerous collaborators that are helping us one by one discovering the new code of this microbe with the immune system. I thank you all for your attention.
Thank you, Yasmine, for a fantastic talk. There is time for a few questions. One question that came to me in the second part of your talk is, if you grow an animal in a perfectly sterile environment, it still develops. Does the gut look completely different, or do you have parts? Or
So in complete absence of microbes, the immune system is slightly underdeveloped, but not as underdeveloped as people thought. But in complete absence of microbe doesn't mean absence of microbial product. An animal that is raised in the condition that they refer as germ-free is still exposed to microbial product via diet and via the beddings. So scientists have actually begun to raise the animal in complete sterility, in which they have also eliminated all the microbial product. And in this case, the animal tend to be really underdeveloped in terms of immune system, and the GI tract is severely affected. So even for the proper development of organs, we need actually this instruction for microbes or microbial product.
Fantastic. So there are questions in the audience. We start with Professor Misha Freismuth, as tradition.
So in your slide, you emphasize the importance of IL-17.
Yeah.
And then, of course, I beg to differ, because you know that IL-17 is the target for psoriasis.
Absolutely.
So, in other words, what is the effect if you would predict that if you take out IL-17 with the monoclonal antibodies, which give a beautiful response in psoriasis, how does that affect the skin microbiome?
Yeah. So, very important point that is exactly in the context of this balance. So under normal circumstances, 17 is actually very physiological cytokines, because actually it's important for barrier integrity, antimicrobial defenses. And you're absolutely correct, an aberrant IL-17 responses underlies very severe inflammatory disorders, not just in the skin, but also in the GI tract or in the brain. So we have actually evidence that in the context of psoriasis, what you develop is aberrant reactivity to the microbiota. And if you actually collect the lymphocytes that are present in the tissue, a large number of them are actually directed against the microbiota. So it is very likely that what we see in the context of severe skin inflammation that are mediated by 17 is in part mediated as an overreaction to a beneficial microbe that tend to be very 17-driven. And in particular, for example, we find enormous amount of responses to staph, which is not surprising, but also to fungi that are naturally present in the skin. So aberrant reactivity to the microbiota is very likely underlying and amplifying a large number of skin inflammatory disorders through this IL-17 axis.
George Tingle, skin immunologist.
A very short general question, then a skin-specific question. The general question would be, when you told us that the microbiota colonize the different tissues and surfaces, are they also there in order to keep their symbionts, let's say keratinocytes, in a state of alertness? In other words, that they are prepared to enhance reactivity?
Yeah.
This was my first short question. The second one, skin-specific. When you mentioned about MHC-like molecules in response to microbiota, do you think that CD1 family, Langerhans cells, CD1A, DDC, CD1C, do you think that they might be involved?
Yes. So the first question is extremely important. I think this is exactly what the microbes are doing. They are creating this kind of tonic activation, and exactly this state of awareness is actually the optimal state for the immune system. You need the system to be in constant training. And the analogy will be a training wheel for the immune system. You really need the immune system to be constantly tickled, constantly educated to be able to launch in the context of an aggression. The point that you're referring about CD1 is exactly true. So we're now actually uncovering a link with HLA-E, CD1 molecules, and you're absolutely correct. A large number of those non-classical MHC, what they do is present very canonical antigen that can be present in numerous microbes. And we think that this is a way by which the immune system is able to deal with this enormous diversity of microbes, is actually using very conserved molecule, non-polymorphic, that can actually allow the immune system to communicate with a very diverse group of microbes. So CD1 molecules are definitely involved in recognition of certain ligands emerging from the microbiota.
So while the students make up their minds and ask the promised questions, does this mean that for each of us, there is a certain ideal interaction with particular bacteria and particular strains in the bacteria, so that our genome is in fact limiting the kind of flora we are able to deal with?
So it's a mutual dialogue. So our genetics is shaping the kind of microbe we live with. Some beautiful work that was done early on by Ruth Ley, for example, showed that actually we are predisposed based on our molecule, on our MHTs or our genetic wiring to have different kind of microbe. But the point that I would like to argue about that is, in high-income country, none of us may have the right microbes. We have done so much to address them. It's always possible. Then I think it will be difficult for us to even understand what is an ideal state. So I think we have probably done a number on our microbiota population and changed in so many ways the way we live, with our environment, our nutrition, our consumption of antibiotics, that we may have depleted the microbe that we actually need. And this, as I mentioned before, may underlie a large number of inflammatory disorders that are in dramatic rise. I mean, most of our children become allergics. We clearly have created a state of imbalance, and it's one of them could be in a relationship with these microbes.
Fantastic. So please, yes?
Thank you. Speaking about allergies and a depleted microbiota, so my question is about whether we get protective immunity from animals, like farm animals or pets. Speaking to American doctors, they've told me they hypothesize that there's this defensive regularis function that comes from animals. Speaking with veterinary biologists, though, they reckon that the effect is through the direct toxicity channel. So that means that we have such a sterile world now in cities, in urban areas, that we need to be exposed to some malignant bacteria from animals in order to exercise our immune systems. And I'm curious whether it's both, one more than the other.
I think it's a complex question, but it is obvious that we have, the way we live in a highly sterile environment is not the way we have evolved. I mean, we did not evolve to live in our highly, sanitized buildings and in very sterilized food. So having actually exposure to farm animal has been shown to protect against allergic disorders. And this could go through numerous ways. One of them could be microbiota sharing. In fact, a lot of the microbe we probably used to have were part of the animals we used to be living away closer proximity. It could be also through the exposure to product, and also it could be exposure to more infection. Although the argument that is developed today, when you compare actually more traditional societies that have been less exposed to in particular antibiotics, they actually tend to have a more diverse microbiota, or tend to be more poor. And I think this rarefication of the diversity of a microbe is believed to be really an unstable state and not a very physiological state for us. So which one is the element that is causative of inflammatory disorders and allergies? Probably many. I think what we have done to our immune system is enormous. We have changed the way we live, we have changed the way we eat, we have changed our microbiota, we have changed our infection. And I think the cumulative cost of all those changes is what we're actually perceiving today in the development of inflammatory disorders.
We have a Mark Smith in the back. Maybe Mark, could you be so kind and stand up so we can see you?
I know you mentioned that the study of the microbiome is a very young field.
Yeah.
However, in your opinion, which approach do you think is most promising for modulating the microbiome to treat disease?
So I think it really depends on the disease. I mean, as I mentioned before, transplant of microbiota is a very valid approach. For example, I was discussing with people earlier about what the work of Eric Paymer is doing in Sloan Kettering in the States. And what he does is actually banking the microbiota of a patient before they give complex antibiotics, and actually repopulating this patient with their own microbiota proved to be extremely efficacious at preventing the negative consequences of antibiotic treatment. So that's a very simple, straightforward approach. People are trying to uncover consortium of microbes that could actually be potentially utilized as a small pill that you can take. But I think what we need also to understand is the ecology. And actually, we need to understand how to change our diet in a way that is actually allowing us to tame those wild creatures that live with us. And I think this is probably one of the most efficacious ways by which we can change our microbiota. The one element that has the most impact on our microbiota composition is our diet. And I think if we begin to actually have a better understanding of which essential nutrients change which kind of microbe, this could offer an enormous, extremely practical approach to actually work with these microbes. The skin is an environment in which we can change also the nutrient landscape in a very efficacious way. That's probably what we already do by applying cream without knowing what we're doing on our immune system. And finally, I think the next approach is going to be the hard one, which is the one in which we need collaboration with chemists, in which we need to uncover the molecules. And that is really, to my opinion, the biggest challenge that we have as a community, but also the most exciting one. Because figuring out the molecules is the key to novel class of adjuvants. So I think the first I mentioned are the ones that are happening right now and are already in trials in many different places. The next one is going to take time because it's going to just be a field of discovery and identification of molecules.
The bottom line is that if you are your age, Mark, and you want to become a billionaire, you can start one of the several companies because there is so much to do and there are so many opportunities to make people healthier and smelling better and being happier.
Or you can stay at the bench and discover the molecules.
Both. Hatoum Basim, another student.
I was wondering if you know whether these microbes are capable of going beyond these barrier sites and kind of finding refuge in storage organs such as adipose tissue and so on, and surviving there in a long term without causing an inflammation.
Yeah, so it has been shown that certain class of microbes live not just outside but are able to colonize tissue. It was shown in the pear patches, for example, where a certain class of member of the microbiota, the natural habitat is within the tissue itself. I don't think it has been shown yet into the adipose tissue, but it is possible that a very subset of this microbe have evolved to live within our own body. But then we already have those endogenous retrovirus, we already have microbial stimulation in our system. I think what is happening now in the field of tumor is actually then people are uncovering microbes that appear to really live within tumor environment, which escape the gut environment or the skin and are able to lodge themselves in the tumor, and they're looking at how those microbes influence in a positive or negative way tumor outcome. But I think this is something that needs to be actually explored in more details. Yeah.
There's a question here.
I'd like to ask if you have the data or if you have the opinion on how the immune recognition of a microbiome constituent species changes when it changes the location of the encounter. in the case of staph epidermidis, for example, if it's on the skin versus if you find it somewhere that it managed to breach the barrier and maybe it's somewhere infecting an internal organ, because it is a big pathogen in hospital infections.
But that's what I was mentioning. Most microbe we have can be dangerous. So a skin microbe is only healthy as long as it stays within the outside of the skin. Staph epidermidis, which is of course in all of us and a very gentle partner, is one of the most dangerous microbes if it actually access catheter in the context of hospital. So when the microbe enter into the immune system, there's no differences between the microbiota enter and the pathogen. It's actually now an aggressor. If it breach actually the barrier, it enter in your bloodstream, it's a pathogen.
So that means that different cells and molecules also recognize it
Absolutely.
in different way.
And the kind of immunity you develop to the same microbe when it enters is vastly different of this gentle homeostatic response.
So an additional feature of the Landsteiner lecture is that we have food and drinks below. And so all of you who have questions for Yasmine, or some of you that have questions for Yasmine, you can ask them now with a glass of wine or a glass of water. And please join me in thanking Yasmine Belkaid for a fantastic lecture. And thank you very much again.